Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Suresh Damodaran. I'm a postdoc at the Duke University and will be moderating today with Dr. Natasha Shylo, a postdoc at Stowers Institute. We are excited to highlight the work of our outstanding postdoctoral members. Today, Dr. Yinan Wan from University of Basel, Switzerland, and Dr. Keng Ioi Vong from UCSD will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box. I'm thrilled to introduce our speaker, Dr. Yinan Wan. Yinan Wan completed her bachelor's degree at Peking University before earning her PhD at the University of Cambridge. During her doctoral research, she worked alongside Dr. William Harris at Cambridge and Dr. Philip Keller at HHMI campus. Her work focused on microscopy and sequencing techniques to explore nervous system development, leading to several published papers. Currently, Dr. Wan is an EMBO postdoctoral fellow at the University of Basel in Switzerland. There, she focuses on spatial transcriptomics and has developed an in situ hybridization method to study whole embryo development in zebrafish. With that, I am pleased to invite Dr. Wan to share her research with us and amaze us with all the cool microscopy images.
Yeah. Thank you, Suresh, for the kind introduction. And thanks for the organizers for giving me this opportunity to present my work. Today, I'll talk about whole embryo spatial temporal transcriptomics. So embryonic development is a process of building complexity in space and time. Here you see a zebrafish embryo developing outside the mother in the first 24 hours of development. So the cells go through cleavage divisions, then through a process called gastrulation, where 3 distinct germ layers are formed. Then different tissues and organ primordiums are generated, all in a very regulated fashion and very rapidly within the first 24 hours of development. This makes zebrafish a perfect model organism to study early embryonic patterning. So a fundamental question in developmental biology is how a pluripotent cell differentiated into different fates. Previous work in the lab has isolated single cells from zebrafish embryos across developmental time points and used their single cell transcriptomics to reconstruct what we call these developmental trajectories to reflect this branching process. However, what is...
Sorry to interrupt. You have a pop-up on your screen. Could you please close that?
Sorry.
Thank you.
How do I do it?
Yeah, just you need to click the X. Yeah.
No, it's not closing. Okay.
Oh, it's gone. Thank you.
Yeah, thank you. Sorry for the interruption. Yeah, so however, what is critically missing in this process is the spatial context of the cells. So cells don't occur in isolation. They go into development in this complex spatial context where they signal to each other genes pattern the embryo spatially, and more importantly, boundaries between the tissues need to form robustly during development. Fundamental questions remain unsolved. What is the full diversity of gene expression patterns? How do gene patterns change over time to establish spatial dynamics? And how do tissue boundaries form? So to answer these questions, we chose 3 developmental stages: early gastrula, mid gastrula, and early organogenesis stage, because they span the lifetime from early embryonic patterning to tissue boundary formation. But the first challenge we encounter is a technical one. How do we quantify, profile many genes in the same embryo? So traditional methods, for example, fluorescent in situ hybridization, can in principle profile transcripts in the entire embryo, but the throughput is limited because the number of genes you can probe usually scales with the number of fluorophores you can put into the system, usually no more than 3 to 5. With the advancement of modern spatial omics method, either based on sequencing or hybridization, it becomes increasingly feasible to profile genes in large scale, but to our knowledge, all of the currently commercially available methods need to section the embryos to 10 micron sections. For zebrafish embryos that live on top of a yolk, if you section it, you only get a ring of cells, which is very hard to reconstruct in 3D and not very efficient use of your imaging space. To address this question, we were determined to develop our own imaging platform to profile gene expressions in thick tissues or whole embryos. We call the method WeMerFish. The method works like this. You have mRNA molecules inside the cells. So we put in DNA probes against mRNA molecules that have acridide modifications. So when we cast the acrylamide gel on top of the sample, the DNA probes get cross-linked very stably into the gel. So we can clear away all the protein and lipid in the sample, so it becomes very transparent. Then we mount the sample onto a spinning disk confocal microscope, which is equipped with a microfluidic system, so we can flow in the linker probes and fluorescent readout probes, followed by chemical stripping of the probes, so only the DNA probes stay in the gel. So we can do this round after round. We can either image a single gene per round, or here we use the MERFISH encoding scheme where we image a different combination of genes in each hybridization round, and based on single molecule colocalization patterns, we can decode which molecule belongs to which gene. For example, if a molecule appears only in round 1 to 8 and N, we know this is a gene 1 transcript. So in this way, we can image many genes in fewer imaging rounds. So in the library, we designed 495 genes that are spatially or temporally variable in our molecular trajectories. We put 471 of them in a MERFISH codebook, and 24 highly expressed genes, we imaged them sequentially to avoid molecular crowding. So additionally, we've developed a branching amplification regime to boost the signal by approximately a factor of 3 to compensate for the signal loss in the confocal imaging. Through these 4 points of technological advancement, we were able to profile genes in deep tissues or whole embryos. Here's what the data looks like. Take the 6 somite stage embryo as an example. We would remove the yolk and flat mount the embryo onto a cover slip. So if here you see an overview of the embryo, if you zoom into a single field of view, you see the tissue architecture is largely preserved. And here is an XZ section of the microscopy image where the scale bar is 10 microns. You can see we can image 120 microns into the tissue without losing much signal. If I further zoom in, you can see 2 genes imaged sequentially, MALD1 in the adexal cell and TBXTA in the notochord. I hope you can appreciate the single molecule signal and the sharp boundary between the 2 tissue types. Additionally, we have oligoconjugated antibodies against the cell membrane, so we can read out the cell membrane signal in the imaging round. So through cell segmentation programs, we can carefully delineate where cell boundaries are, and if we further zoom in, we can assign these single molecules to individual cells or even subcellular compartments. Here's what the data looks like. I'm showing you a compilation of 495 genes imaged across 3 developmental stages. We have 2 replicate embryos per stage. You can see the consistency between the replicate embryos very good, and I hope you can appreciate that diverse gene expression patterns in this rich data set. So it may look like 500 in situ hybridizations, but bear in mind, here in every single cell, we have 500 genes profiled simultaneously, and across the 6 embryos, we have over 130,000 cells in there. So like I said, we measured 495 genes expression patterns. In the lab, we also have single-cell multi-omics data, which means single-cell RNA-seq data that profile the RNA expression pattern and single-cell ATAC-seq in the same cells with the chromatin accessibility patterns. So through an algorithm called Tangram, we were able to map where these single cells come from in our reference atlas, and we're able to impute the rest of the genome as well as nearly 300,000 chromatin accessible regions. Here I'm showing you a gallery of the measured gene expression patterns in red, imputed patterns in blue, and imputed ATAC peaks in purple. So again, this is a very information-rich data set. We move on to the questions we're interested in answering. So we've also made this into a browsable website where you can check your favorite gene, check their localization patterns. And there is a UMap on the corner of the website where you can hover onto it and check which cell type the gene is expressed in. And on top of the web page, there is a genome browser where you can click on the chromatin accessibility region and to check the specific ATAC peak and how the accessibility pattern is imputed in space. This would be particularly helpful if, for example, you are a biologist wanting to design a reporter assay, for example, for your regulatory element. So as I said, Murphy measured spatial patterns of 495 genes at subcellular resolution. Integration with single-cell multi-omics data generated an atlas available at Murphy Eyes website. So we move on to our questions. How do gene patterns change over time to establish spatial dynamics? What I mean by spatial dynamics is that although development happens in time, it also happens in space. A lot of time, developmental time is recapitulated in space. So here you see a beautiful movie of the zebrafish somatogenesis with the somites generated one after another from the anterior to posterior axis. If you draw an arrow from the posterior end to anterior end of the body axis, you transverse through time. You go through the stem cells to progenitor cells to more mature cell types. This is indeed what we found. If we map the molecular trajectory that lead to the somite stage, and we estimate where they come from in these three stages combined, we found that these cells trace back to the margin cells at 50% epipely, to non-dorsal margin and 75% epipely, and there is a posterior to anterior cell fate progression maturation gradient happening in the 6 somite stage. We verified this with an independent analysis called a modified version of RNA velocity analysis. If you're familiar with the traditional RNA velocity analysis, you know it uses spliced and unspliced RNA count to estimate the temporal dynamics of gene expression. So here, instead of spliced and unspliced count, we used nuclear versus cytosolic count. So transcription happens inside the nucleus. This means if you see an RNA molecule inside the nucleus, it reflects an early fate of the RNA molecule and the gene is being upregulated. Conversely, if you see an RNA molecule inside the cytosol, it means it might be in the way of being degraded and the gene is being downregulated. We use the fate diagram of the RNA velocity to estimate where in the next time point the temporal gene expression pattern is going. And we were able to reconstruct these trajectories in space where this is the tail of the zebrafish. We recapitulated a bifurcation process where on one hand, the progenitor cells are going down to the spinal cord trajectory, on the other hand is going down to the somite region. So we've identified the basal or more ancestral stage to this cluster, which is located at the dorsal core of the ectoderm tail bud. So this is consistent with the literature report of the existence of the neuromesodermal progenitor cells in this region. And more importantly, RNA velocity recapitulated this posterior to anterior pattern maturation gradient. So what's surprising is when we applied RNA velocity analysis to early embryonic development, strikingly, we found that the RNA velocity stream mimic how the cells would move. Here you see the bulk cell movement patterns in early embryos. You can see the margin cells moving to the animal pool, and there is convergence extension going on in the hypoblast. So we were quite surprised by this observation because it's not very obvious that the dynamic expression of genes that are driving the cell movement. But if you think about it, it makes sense because suppose if you are a cell close to a morphogen gradient source, you want to upregulate your target gene because you're even moving closer to that source in the next moment. But there's really unexpected link between the temporal dynamics of gene expression and morphogenetic movement primed us for many interesting directions in the future to probe this link. So temporal gene expression aligns with cellular maturation and morphogenetic movements. Finally, we're going to probe the question of how do tissue boundaries form. So in early development, gene expression happens in gradients, and in the early organogenesis stage, the cell boundaries, tissue boundaries have already formed. So we focus on mid-gastrular stage to probe where cell type boundaries are and how they emerge during development. We took an objective approach where we took a local neighborhood cells and evaluate how spread out their gene expression patterns are in the gene expression space. The hypothesis is that at the tissue boundary where there are multiple cell types in the local neighborhood, the heterogeneity score would be high. Indeed, this is what we found. The sharpest cell type boundary happens in this stage in the mesoderm between the axial and paraxial mesoderm. If we evaluate the gene expression pattern across these sharp boundary between the notochord region and somite region, here x-axis is distance to the boundary and y-axis are genes, you see that we very sharply transition from one group of gene to another group of gene. So how is this boundary formed during development? So literature told us there are two ways of sharp boundary formation if it is challenged by cell proliferation or tissue deformation or just this noisy signaling environment. Cells could either switch their identity and adopt the fate of their neighbor cells, or they could move to the target location through cell sorting to form a straight boundary. Which case is this for our notochord and somite boundary? So we took a step back and look at 50% epiploid stage. To our surprise, except a small cluster of genes expressed at the dorsal margin, most of the genes are very broadly expressed across the dorsal ventral axis across this boundary, indicating there is a large-scale cell state remodeling during the formation of this boundary. What about cell behaviors? So we used light sheet microscope to quantify the cell behaviors during this process, where we tracked all the cells and identified their fate based on the transgenic marker MIF5 to identify which ones are somatic mesoderm cells, which are notochord cells, and we tracked them back to their developmental origin. To our surprise, this is an amazingly orderly process. Here you see the quantification of cells distance to the boundary with the identity of cells and progression through time. If we take snapshots across this process, you see that the red cells mostly stay on one side of the boundary and blue cells stay on the other side of the boundary. There is very little cell mixing going on. So this further confirms that the formation of this boundary is through cell state change rather than cell sorting. So sharp cell type boundaries form at mid-gastrular stage at the notochord somite border. Cell state change rather than cell sorting generated this sharp boundary. So this concludes what I want to say about our resource and findings. I just want to emphasize that Wimmerfish is not only applicable to early embryos, it's a generalizable method for thick or whole tissue spatial transcriptomics. If you have questions about the method or the data, please check out our bioRxiv paper. And shout out to the zebrafish community out there to check out the Murfish Eyes website. We hope it's a good complementary to the ZFIN in situ database. Finally, I just want to thank all the people involved in this project. This has been a wonderful collaboration between our lab and Bogdan Bintu's lab at UCSD. Jacob, a graduate student in the lab, and I co-developed the Wimmerfish method. And KJ is a very talented undergrad student in Bogdan's lab who developed the Murfish Eyes website. Thank you.
Thank you, Yinan. That was really fantastic. now we are starting to get questions. So the first question is from Natalie Hussein. So she says, fantastic presentation, thank you. Has this merfish technique being applied to other species like chicken embryo?
So far, I have not seen this being applied to chicken embryos. There is another method using slightly different strategies in mouse embryos for deep tissues, but I imagine this can be applied to other species as well.
That's great. I think more questions are going to come up. I have a question. So with regards to genes that are like poorly expressed that doesn't have like enough RNA, how does the in situ hybridization approach works for that one? Like in terms of like gene expression levels, does the genes that are like poorly expressed, are you able to still probe them?
Sorry, can you repeat the question?
For like genes that are like poorly expressed, are the probes still be able to detect those genes?
Yes. You mean along the RNA velocity trajectories?
Yes.
Yes. So you can see this like early stage of progenitor states happens like in the tail bud, and it comes out of the tail bud and move to the corresponding regions to the spinal cord or the somatic trajectories.
Great. We have a next question from Grant McGregor. Wonderful presentation, thanks. What is the average number of transcripts counted per cell?
I don't remember this on top of my head, but we have millions of transcripts in total. We've compared Murfish method with, because in our probe design, we can do both sequential fish, which is basically single molecule fish. We have compared with the single molecule fish with combinatorial encoding. And we found out that we could recover between 60 to 90% of the transcripts in the cells. If you consider the SM fish as the gold standard of the field, so that's how much we can recover.
Okay, great. Still waiting for a few more questions. There's a new question from Chen Kao. Fantastic talk. Does the merfish have efficiency differences between tissues in the surface or inside the embryo, like looking at the 3D level? So
not really. Because of the tissue clearing, we made the sample super transparent. If you haven't noticed, the two embryos I showed at the 6 somite stage has been, like, I accidentally mounted one dorsal side up, one ventral side up. The consistency between the two embryos is really good, and you can't even tell the difference between which side the embryo is imaged.
okay, great. While we are waiting for other questions, I also want to know, like, what is the specificity of using the spinning disc confocal for your fish, like for the microscopy? You mentioned that you are using the spinning disc confocal.
Yeah.
Is there a specific reason that you need the spinning disc confocal for this?
Yeah, so there is indeed a little bit tissue background, although we've done clearing. So with a regular wide field microscope, we can't get through more than a couple of layers of cells. But the pinhole in the spinning disc confocal microscope really rejects autofocus light and make the sample really clear deep in the tissue.
Okay, great. We have another question from Rasik Takal. Wonderful presentation, thank you. A quick question about the RNA direction tracing. Is it tracked using probe and traced in real time, or is it fixed in different stages?
It's fixed. Yeah, so like I said, RNA velocity is a mathematical method to estimate the direction of transcription dynamics. So like I showed here, is really using the count inside the nucleus and cytoplasm to it's an estimation of where the gene expression pattern is going rather than live tracking.
Great. There's another question from Chris Wright. Can you determine the absolute percentage efficiency of counting Wimmerfish transcript numbers, counting the dots, compared to the real number of RNAs in the cell by single cell analysis or something?
Yeah, I mean, I think there is still debate in the field what is really the gold standard of counting the number of transcript in the cell. So we've actually compared our method to because we have every single transcript in the embryo. We've compared our data with bulk RNA-seq, and the correlation coefficient is about 0.6, which is quite reasonable. But we also argue that maybe bulk is better than any single cell sequencing method, has a very high dropout rate, and it's hard to say that's the real count of the RNA number in the cells, right?
Yeah. Okay, so I think that's all the questions that we have. Thank you, Yinan, for the great presentation. It was really great having you.
Thank you.
Thank you, Nan. That was a really great talk. Hi, my name is Natasha Shylo. I'm a postdoc at Stowers Institute for Medical Research, and it is my pleasure to introduce Keng Ioi Vong. Dr. Vong did his PhD at the Chinese University of Hong Kong with Dr. Kim Kwan, and is now a postdoctoral researcher in the lab of Dr. Joseph Gleason at University of California, San Diego. He's interested in how genetic variations affect the development of nervous system. He has an impressive publication list and numerous accomplishments, but I particularly want to highlight the Young Investigation Award at the 7th International Enteric Nervous System Developmental Meeting. And with that, let's hear his excellent talk, please.
So thank you for the amazing introduction, and I'm very happy to share with you my recent work on how to use somatic mutation to reconstruct the lineage of trunk neural crest-derived sensory and sympathetic ganglia. So most of the peripheral nervous system, including the sensory, the sympathetic, and enteric neurons, they are derived from the neural crest cells. So neural crest cells, they are progenitors derived from the roof plate of the neural tube that undergo epithelial to mesenchymal transition to migrate out of the neural tube in a process that we call delamination. So following their delamination from the neural crest, they will migrate to their destination, including the dorsal root ganglia, the DRG, and also the sympathetic ganglia. So the migration paths are already well defined, but the lineage relationships of these different neural crest derivatives are still unclear. So specifically, there are two competing models of neural crest cell fate specification. So the first model suggested that the delaminated neural crest cells, they have the multipotency to generate different cell types, while a second model suggested that the cell fate specification already occurred when the neural crest cells, they are still in the neural tube, and the delaminated neural crest cells, they generate predominantly a single cell lineage. So over the years, there are already a lot of amazing scientists that contributed to these two different models. But what is unclear is that which of these models may represent the predominant mechanism during in vivo development. So why is this question important, and why should we understand the lineage of neural crest cells? So first of all, the lineage organization is the blueprint of building an organ. And with this blueprint, we can learn how progenitors diverge into specialized cell types and how these different cell types can be organized into a functional tissue. Now, in some cases, there are somatic mutations that are acquired during development instead of inherited from their parents. So when a progenitor cell divides, any mutation will get inherited by all the daughter cells, creating what we call a clonal populations. So the result is that only the cells in these specific clones will have the phenotypic consequence of that mutations. So take a look at this brain of a disease called focal cortical dysplasia, which is a somatic brain disease. So you can see the abnormal tissue is sometimes restricted to a very specific loop or a very specific brain region. But in some cases, the mutation is very widespread in the entire hemispheres. So understanding the lineage will tell us why some cells might be particularly vulnerable to somatic mutations, and why sometimes only certain cells will have the phenotype. However, the conventional lineage tracing tools, they have some limitations. So for example, in the confetti mouse model, it doesn't have more than 10 combinations of fluorescence. And this limits the resolution that we can use to resolve the lineage relationships of different cell types. So the ideal case for a lineage tracing tool is that we can assign a unique barcode to each cell in the body. So how can we do this? So one way to do this is to use the somatic mutations. So as I mentioned just now, somatic mutations, they are acquired in almost every cell division. So you can imagine not any two cells in your body, they are genetically identical. So that the whole body is actually a genomic mosaic. And because these mutations, they are always, like almost always accurately inherited in all the daughter cells. So so any shared mutations in two cells would imply that they are derived from a common ancestor. So most of these mutations, they are neutral and they are not going to cause any disease. So by analyzing these mutations, we can reconstruct the clonal relationship and the lineage structure of an organism. So let me briefly talk about how we can use somatic mutations as cellular barcode to trace lineage. So an important concept in doing this is allele fraction. So allele fraction measures the percentage of cells in a tissue that have a certain somatic mutations. So let's say in this DRG here, so half of the 20 cells, like 10 of these 20 cells, they have this mutation, which is this orange circle. So the allele fraction will be 0.25 because all these cells are diploid and only one copy of the DNA will have this mutation. And consider here, like this DRG, only 2 of the 20 cells have this mutation, then the allele fraction will be 0.05. And if the DRG doesn't have any of these somatic mutations, then of course the allele fraction will be 0. So by calculating the allele fraction for many different mutations, we can tell how similar are the clones in different tissues. So let's take a look at this example where there are 4 different somatic mutations. So by comparing the allele fraction here for each mutation in each of the different tissues, so we can infer which of the ganglia are more clonally related. And in this example, because the T1 DRG and the T2 DRG, they share a more similar mutational pattern, then we can tell they are more related in terms of their lineage. So based on this rationale, I'm going to deconvolve the lineage of the sensory and sympathetic nervous system, which are derived from the trunk neural quest cells. So we'll answer 2 questions. So first, are the laminated neural quest cells multipotent? And the second is, what are the lineage relationship of all the sensory and sympathetic ganglia in the body? So we first answer the questions using mouse models. So although we can use the intrinsic somatic mutations in the mice, there's a more efficient way to do this, which is to use the CRISPR-Cas9 system to artificially create some somatic mutations in the neuron quest lineage. So the way we can do this is to use a homing guide RNA mouse model, where the guide RNA will target itself, and then we can use a neuron quest specific Cas9, which we can do it by using a source tanquy, which specifically restrict the Cas9 activity in the delaminated neural quest cell lineage. So here we can see 2 examples of the barcodes. So each barcode can have many, many different mutations because the Cas9 edits are random. But the mutations, they're all inherited in the progenies. So we cluster all the different ganglia by the allele fraction of each of the different mutations. And we can see the blues, which represent the sympathetic ganglia, they are clustered with each other, and the red, which represent the DRG, they are clustered with each other. So you can see a large chunk of blue and also a large chunk of red. So when we take into consideration more than 20 different barcodes and more than 200 mutations in total, we can still see a very similar pattern, which is the blue and the red, they cluster independently. So we can take a look at these 2 example clusters. So when we map the components of these clusters to the anatomy, so we can see that the ganglia in these clusters are mostly made up of one type of ganglia. So this is either the DRG or the sympathetic ganglia. So this may imply that these 2 tissues, they are derived from 2 independent lineage, in the mouse. So, but the next question is, how about in humans? So for humans, we can't introduce mutation by CRISPR-Cas9. So we will have to analyze the inherent somatic mutations. So we collected 3 independent neurotypical adult donors. We dissected the control organs, and we dissected also the DRG and the sympathetic ganglia. And we extracted the DNA and put them to whole genome sequencing to identify and validate the somatic mutations. So we are able to identify several hundreds of these different somatic mutations, which we can use as a cellular barcode to reconstruct the phylogenetic lineage relationships of these different tissues. So based on these hundreds of mutations, we could reconstruct a lineage tree of the DRG and the sympathetic ganglia. And because there are so many different ganglias in the body, I'm not going into details like how all these they are related to each other. But again, we can take a look at some examples. Like in these 2 examples, the clusters of like the green clusters and the purple clusters, each of these clusters, so the components are mainly made up of one type of ganglia. So either DRG or sympathetic ganglia. So this would suggest that the DRG and sympathetic ganglia, they are likely derived from distinct progenitor pools. So another thing we observe is that the clones, they are organized along the rostrocaudal axis, which means that they are like distributed along many different axonal levels, but not between the sensory and sympathetic cells of the same level. So, so what can explain this spatial organization of these clones along the rostrocaudal axis? So there could be 2 possible reasons. The first one is because of the migration of the DRG and sympathetic cells, like between all the different ganglia. And second is it possible that the neural crest cells, they can migrate across different axonal levels when they're still within the neural tube. So we are going to examine like which of these scenarios like actually drive the clonal organization that we observed. So what we have been doing is we map the mutation allele fraction onto like this body plan of anatomy. And then from these examples, we can see that many of these mutations, they actually distributed very like across many different levels, from all the way from T12 to the cervical ganglia. And then most of these mutations, they are actually distributed to both sides. So this seems that it is unlikely the clonal organization that we observe is because of the migration of individual ganglia cells that span across so many levels. So instead, we take a look whether neural crest cells, they can actually migrate between different axonal levels when they're still within the neural tube. So we collaborate with Dr. Melanie White in the University of Queensland to do a live imaging for these movements of neural crest cells in the cryo embryos. So we electroporate a neural crest cell reporter into the early stage cryo embryo, and then we live image the movements in the 6 hours. So we can take a look at this video and see how this neural crest moves. So it's not working, but I can do it manually. So you can see, so this is the green cells that are the neural crest, and then we have also done some tracking. You can observe the path of these neural crest cells. And what you can observe is that they are migrating along the rostrocaudal axis between different axial levels. And these are some examples of the tracked paths that we have made. So the result is that when we quantify like how long these cells migrate, so within the 6 hours that we can image, more than 20% of neural crest cells, they can migrate more than 80 microns, which is more than 2 somatic levels. So this suggests that neural crest cells, they can migrate along the rostrocaudal axis when they are still within the neural tube before the delamination. And this migration behavior is very likely contributing to the clonal organization that we observe by distributing the clones to different axonal levels. Then we ask a question, so is it possible that a specific cell type drives the rostrocaudal clonal organization? So we decided to look into the clonal relationship at a single cell resolution. So we first isolated 220 single nuclei from the DRG and also the sympathetic ganglia from the T2 and T3 levels. Then we use a novel approach called Resolve-Own, which can profile the DNA and RNA from the same nucleus. So DNA is used for genotype to somatic mutations, so it can tell us like what mutations are there in the nucleus. And RNA is used for identifying the cell type. So this way we can generate a cell type resolved phylogenetic tree for all these 200 nuclei. So this is the actual phylogenetic tree, but we don't have to go into the details here because there's just too much information inside. But let me show you what this map can tell you. So first we can see that the clonal organization is not driven by either neurons or glial cells, because you can see that both of these cell types, they get intermixed with each other. And then when we look into the terminal branches, so terminal branch would mean that a pair of cells that is most clonally related in this map. So we can see that most terminal branches are formed by the same type of ganglia in the same level. So this is kind of expected because the progenitors in the ganglia, they may undergo clonal expansion within the tissue. But however, if we focus on the type of terminal branch that consists of different types of ganglia, that is, they consist of a pair of DRG and sympathetic ganglia, but they are coming from the same level. So we can actually see that the percentage of this type of terminal branch is less than expected by chance. So this would imply that the same progenitor is less likely to generate both DRG and sympathetic cells. Then the question is, how do we test this hypothesis? So just like in a crime scene, every contact leaves a trace. And I was saying in development, every mutation also leaves a trace. So although we were not able to see how these cells develop in real time, we can use the somatic mutations as a mark to reconstruct what happens during development. So take the neural crest as an example. When the neural crest cells, they delaminate from the neural tube, it either goes to the left or goes to the right. It might go back to the opposite direction, but it is like kind of rare. So you can imagine the left or right decision is actually marking the moment when the cell leaves the neural tube. So any mutation that's on one side, it is likely that it is acquired after the cell leaves the neural tube. So how do we use this information to reconstruct the different events and answer the questions that whether the delaminated neural crest cells, they are multipotent? So let me give you an example here. So consider two types of somatic mutation. One is acquired after delamination, and the other is acquired before delaminations. So, and then let's consider what we could observe when neural crest cells are actually multipotent. So for any mutation that are acquired after delamination in this scenario, so in the case, if they are multipotent, then of course they can generate both DRG and sympathetic ganglia. So you will see those mutations on one side, but in both types of tissues. But if the delaminated neural crest cells that are phase specified, then you would expect to see them on one side and also in only one type of ganglion. And for the mutation that is acquired before the delamination is quite similar, but the only difference is that this mutation, they will be distributed to both sides, but depending on whether the neural crest cells that are multipotent, they will be found in one or two types of the ganglia. So when we take into considerations the several hundreds of different mutations, and if you plot the allele fraction difference between the two types of ganglia against that between the left and right, we would expect a very different distribution in these two scenarios. So what we can do is we can compare the expected outcomes of these two scenarios with what we actually observe here. So, and we can see that the majority of somatic mutations follow the pattern that delaminated neural crest cells are phase specified within the neural tube. So let me give you another example of how we can use mathematical modeling of these somatic mutations to test this hypothesis. So imagine the neural crest cells, they are some marbles in this jar, and the marbles are magical, so they can replicate themselves over time and multiply by numbers. And at some point, the marble will get painted with green, and they keep on replicating. So the green is like when a cell gets somatic mutations. But if we separate the marbles into two separate jars at different time points, the earlier we separate them, the larger the difference will be, the number of green marbles between the two jars. So now you can consider the separation of these marbles is like a developmental decision. It's like when the neural crest cells, they decide to become DRG or sympathetic ganglia, or when they become committed to the left or right side. So, but the issue is we are not sure how many marbles are there in the jar when the separation occurs. So to figure this out, we will simulate different scenarios. So what if there are 0, 10, or 12, like up to like 10,000 marbles in the jar when they get separated? So the thing that we can do is each of these numbers represent a hypothesis of the initial starting cell population size when they separate. Then we will see which number does not fit the actual observation of the allele fraction difference between the two groups. And we can take a look at these two examples. So here we can see that at the smaller number of the cells at the moment of separation, the larger the allele fraction we will observe. Larger the difference in the allele fraction between two different groups that we will observe. So so using this approach, we can estimate like how many cells are there in the population when the progenitor pool separates to DRG and sympathetic ganglia, and also how many cells are there in the progenitor pool when they can separate to the left and right side. So based on this rationale, we can compare whether cell fate decision to become a DRG or sympathetic ganglia happens first, or the decision to become left or right happens first. So we can see like in these two scenarios, so both of these results suggested consistently that the developmental decision for becoming DRG or sympathetic ganglia happens earlier than the decision to become left or right. In other words, so this would suggest that neural crest progenitors, they already have their fate decided when they migrate out of the neural tube. So to summarize these findings, neural crest progenitors, they can migrate along the rostral axis to distribute the clones across multiple axonal levels. But most of the times, these clones, they are not shared in both the DRG and the sympathetic ganglia. Instead, you can see that the DRG of different levels share the same or similar mutations. So this suggests a model that most neural crest cells, they already have their fate restricted before leaving the neural tube. But our single cell analysis also suggests that there's a small fraction of progenitors have the potential to generate both cell types. So I would like to thank my postdoc mentor, Dr. Joe Gleason at UCSD, and I would also like to thank my amazing colleague in the Gleason Lab and also the amazing collaborators that contribute to this project. And we are also very grateful for all the funding sources that make this project possible. Thank you so much.
Thank you. That was an amazing talk. So this would be a great time for everybody to post their questions into the Q&A box. And I guess I will get started with some probably pretty fundamental questions. So when you look at your somatic mutations, do you because I imagine you guys probably have spent a lot of time looking at both on mouse and humans. Do you see kind of specific mutation hotspots perhaps in the genome? Or do you see, I guess it would be a second question, do you see synonymous mutation, like same mutation popping up in lineages that makes no sense for that mutation to be there? And I guess, or do you kind of focus on very specific regions of genome by default?
Yeah, so thank you for the question. This is a very good question. So your question is like whether we see some specific mutations that are like enriched in a certain region or enriched in a certain domain of the chromosome, right? So we do not observe these scenarios. So instead, so for the mouse, because we are restricting the mutation to a certain barcode locus, so of course we do not see those happening in the mouse. But in the humans, so it seems that most of these somatic mutations are actually like synonymous, as you said. And one thing that we actually have done is actually we exclude all those mutations that have a high allele frequency in the database. So those are probably not related to the normal development, right? So this is just reflecting those mutations, like not mutations, but variants in the normal population. So we have a cut-off criteria for those variants that we observe, because that may not reflect the development trajectories in an individual, but instead it may just be some like normal benign variants that is often observed in the populations.
Okay. So we're still waiting for some more questions. So I guess it's my opportunity to ask all of my questions. Do you, I wasn't entirely clear because a little bit of your talk talked a little bit about kind of spatial positioning of some of the more specific, the DRGs and SGs. Can you kind of explain a little bit like how did you know where spatially they were located, and did is there room for like a spatial transcriptomics, in this approach?
Yeah, so yeah, this is a good question. So we have a very good collaborator at UCSD from the Department of Anatomy. So we collaborate with this anatomist to dissect the ganglia from the human donors. So we can have document for like each of where like all these different biopsies coming from. So we can tell. So this is from the T1 level. This is from the T2 level. So for the second question about spatial transcriptomics, actually we are also doing this, but not in the humans, but in the mouse. So we have like a our CRISPR barcode mouse that we can transcribe those barcode, and then we can like map it back to the slide, so we can tell which cell types have these certain mutations and what are those different cell types. So we can see the lineage relationship at like a single cell level, and also with the spatial organization of this different lineage.
That's very cool. The question I always ask about neural crest cell talks, because I'm like just curious. Do you, now that it sounds like you maybe peeked on that data a little bit, do you ever or often see neural crest or progenitors, or descendants of neural crest cells crossing the midline and going over to the other side? Like, do you see any evidence of that happening?
Yeah, that's a great question. So actually, we have done this live imaging in the cryo embryos, but I did not show here because of the only it's 20 minutes talk. But what we have done is we electroporate a neural crest reporter to one side of the cryo embryo, and then we visually inspect whether it's really restricted into one side, and then we image this how this neural crest migrate after the electroporation. And then we observe like around 30 to 40% of these neural crest cells, they can actually migrate across the midline before the delamination.
That's a
Yeah,
lot
this
higher
can-
than I thought it would be.
Yeah, that's pretty cool.
Sorry, I gotta write it down. All right, so we have a question in the Q&A. This comes from Sophie Astrov. It says, nice talk. How do you explain that neural crest cells can be transplanted and adopt a different cell fate than the fate from their original location?
That is a really great question. So I would say that so when you graft certain neural crest cells and then to another location, and then they can like change the fate to like change the fate to a different cell type, right? But probably like when you do this kind of migration grafting, so those neural crests, they are not have their fate specified yet. They do not yet leave the neural tube. They are still within the neural tube. So it's possible that most of these neural crest cells, they do not have the fate specified yet. But what we find here is like when the cells delaminate, they probably have the fate specified. So if you take some delaminated neural crest cells and then try to graft those, then probably this is not going to work. But if you take the neural crest cells that is within the neural tube, then probably like they can still change the fate.
That's cool. There's another question from Billy Swalla. She says, very interesting talk, Keng. What a clever way to see the fates of the progenitor neural crest cells. Do you think that these results will hold true for all vertebrates?
Yeah, thank you for the question. This is a really good question. So I can't for sure say it will hold true for all the vertebrates, but we have taken a look at the mouse and also the humans. So it seems that for the mouse, we use like a SOX10 query, which is specific for all the delaminated neural crest cells. And we see this pattern that the DRG and the SG, they are kind of independent. And in the human, we use an unbiased approach to see all the somatic mutations. It seems the results for human and mice, they are pretty consistent, but so I would say it suggests it might be evolutionarily conserved, but I can't be 100% sure for that.
Interesting, yeah. I wonder if looking at like, some of those animals that just barely have neural crest cells, like landslide or something like that, like whether that would hold true as well. That would be, yeah, it would be interesting.
Yeah, that would be very interesting.
Yeah. All right, I have one more last question. You talked a little bit about like computational and truly tracing the lineages, but I kept thinking can you really get, like, how precise can you get? Can you basically like count the generations of cells based on the number of mutation, and then whether that, whether you can relate that to, times of cell division and convert it into actual time and say this cell, the progenitor cell existed like 2 days ago. That means we now know the progenitor was truly, existed in mouse at embryonic day, I don't know, 7.5 or things like that. Like, is that a resolution that you might be able to do, or for humans even?
Yeah, this is a really cool idea. I think like for now we don't have an algorithm to actually tell like the timing of all these somatic mutations and like when exactly they get these mutations. But I think this is a really good way to go. Like we yeah, thank you for these suggestions. Yeah.
Of course, of course. All right. Well, yeah, well, it looks like that's yeah. Oh, sorry. Billy has a comment in response to her previous question. She says thank you, and she's like, well, maybe it is conserved at least for mammals. So yeah, um, which I think would be very, yeah. Basically, your data is very solid on that. I think unless there's one more question pops up right away, I think we might be kind of done with our questions. Thank you so much for an excellent talk. I want to thank Yinan and Wan and Keng Ioi Vong for your excellent talks overall. This seminar has been recorded and will be available on the SDB website next week. Please join us for next month's seminar on Friday, March 14th, 2025, when Joshua York from Northwestern University and Laura Reck Sessionell from Children's National Hospital will present. Thank you all for coming.